I am at the Bluff Street Bridge in Wayton, and this is where I hope to catch the jellies that are carrying the planula that I'm going to use for my experiment. It's a really good location because on my right here is an enclosed estuary, and at high and low tide, all the water that goes in and out of this estuary needs to pass under this bridge. So I can just stand there and scoop jellies out as they pass by, and when the time is right, when the rain's right, I can catch a ton of jellies that way. The tools that I'm going to use are a nice long net on a pole. It's probably not the best way to catch jellies and keep them intact, but I don't really care about the jellies themselves. I'm just doing it to get the larvae out of them. And once I catch them, I'm going to put them into this 5-gallon bucket and leave them there and let the planula leave the tentacles of the adults, and then I can just collect them off the bottom of the bucket later and settle them out. After the jellies have been sitting in the bucket for a little while, I'm going to bring them here. This is the lab at the Maritime Aquarium, and it has tools that I can use to make sure that the jellies have planula and make sure that I have enough planula to do my experiments. Things like just a pipette, and in the back there's a dissecting scope. You can see the planula with your naked eye, but it can be really, really hard. They're very, very small, and in order to verify that the planula are there and that they're alive, I really should check under the dissecting scope first. After I've collected the planula, I won't really need the medusas anymore, so I will either re-release them back into the sound, or else I can donate them to the Maritime Aquarium, where they can be used for one of the
I will settle out the planula using the methods described in the 2010 paper by Holst and Jarms. This is a caption from that paper. Basically, I will fill the top half of a petri dish with clean filtered seawater from the aquarium that's been adjusted to match the salinity of the planula. Then I will add 1 pipette of planula to the petri dish and place the bottom of the petri dish so it is floating on the top half of the dish. This encourages the planula to settle onto the underside of the dish because they like to settle onto the undersides of objects.
In my experience in the past, planula have actually gone straight into the cyst stage and haven't formed polyps. That's probably going to happen this time around too. So what I've done in the past is I've taken the cysts that are on the petri dishes and placed them in a tank of filtered seawater until they hatch out. I'm going to do that here at the Maritime Aquarium. This is the jelly culturing lab, and I have this tank available to do that. I'll keep them in little containers just like these ones here. These are actually polyps that I've used in the past. They'll be kept on the petri dishes hanging in filtered seawater. It'll be 16 degrees Celsius, and the salinity will be between 28 and 30 parts per thousand. And I'll feed them Artemia nauplii twice a day once they hatch out, and I'll water change them about twice a week and clean around the polyps every time I water change them. Once enough polyps have hatched out from their cysts and have grown to be large enough, I can remove them off of the petri dishes in these containers and start placing them on the petri dishes that I'm going to be using for the experiments. After the polyps have hatched out at the Maritime Aquarium and have gotten large enough, I can transfer them to the petri dishes where they will be run through their experiments. I'll want to transfer about 8 polyps per petri dish because in the studies that I've read, that's the minimum number of polyps that people have used to get somewhat significant results. So I'll transfer the polyps to the petri dish and just let them settle for maybe a week or two, just by letting them sit in filtered seawater for a bit. Once the polyps have attached to their new home, I'll make little markings on the petri dish. I'll make little markings on the petri dish by circling them with a dark marker and then putting little dots around them so that I can tell which one is which. 1 dot for one polyp, 2 for the next, etc., etc. So each petri dish will represent 1 replicate. And I'm going to do 3 replicates per treatment. And treatments will be done in a Rubbermaid container. This Rubbermaid container will be kept at a specific salinity, and I'm going to be doing 3 different salinities. So each treatment will be housed in a Rubbermaid container, placed inside an opaque bucket like this, with a lid. The opaque bucket will keep out any light and inhibit algae growth so that nothing else affects the animals. I don't want light to be a factor in this experiment. These buckets can also be wrapped in a thick opaque opaque plastic like garbage bag. This is white, but it's just here to show what it would look like. I'm going to use a black garbage bag for the real thing. And then place in a tank of filtered seawater held at a specific salinity. So I can fit 3 of these Rubbermaid containers in a bucket, allowing me to test 3 salinities at each temperature. And I will have 3 different buckets so that I can test each salinity at 3 different temperatures for a total of 9 different treatments. Before I start the experiments, I will chill the polyps down by about 6 degrees for a week or 2, because this has been shown to help prime the polyps for reproduction. It's something similar to what they experience in the wild when the temperatures drop in the winter. So as I said, I will be testing polyps at 3 different temperature and 3 different salinity regimes. I will be doing temperatures of 5, 13, and 22 degrees Celsius, based on NOAA records that showed that Long Island Sound can experience temperature fluctuations ranging from 5 degrees Celsius to 22 degrees Celsius. This is a very wide range, and polyps might strobilate at more specific temperatures, but I want to make sure that I'm exposing polyps to a wide range of temperatures so that I can see reproduction in the form of strobilation, budding, and possibly cyst formation. The salinities that I'm going to be using will be 10, 20, and 30 parts per thousand, just based on personal experience of finding animals at those different salinities in Long Island Sound. I will be running the temperature and the salinity experiments at the same time in the same containers as I explained, because temperature and salinity are complementary parameters in the asexual reproduction of polyps. I'll make sure to control for lighting and food during this experiment so that it's not a factor in my results. I'm going to control for lighting by blocking it out entirely. I'm going to house the polyps in the opaque buckets that I mentioned and wrap them in an additional layer of opaque material, the black garbage bag, so that no light gets in. For food, I'll just make sure that each container is being offered as much the exact same amount of food as possible by measuring out the nuclei in a small syringe and giving that same amount to each container. I could even take the syringe of nuclei and freeze it so that later I can analyze it and even count the individual nuclei in the syringe and make sure that it's approximately the same. During the experiments, I will monitor the polyps frequently, checking on them at least 2 or 3 times a week. This way I can feed them, clean them, and check to see if there's any asexual reproduction. I'll note whatever I see, and I will also make sure to note the type of reproduction, if it's budding or if it's strobilation, and I'll even try to note which individual polyp is strobilating. Oftentimes the polyps prepare for strobilation by becoming very long, so it's possible for me to tell if I find a fire in a container which polyp it came from.